Okay. So till now we have discussed the entry inhibitors. So under the entry inhibitors, we have discussed this one, that is CCR5 receptor antagonist. Okay. And then we have discussed how the CCR receptor antagonist will work. Next one, uncoating inhibitors. I've explained you what do you mean by uncoating. Okay, uncoating. Then how these examples we have discussed, that is amantadine and rimantadine. These two drugs, they inhibit the M2 porins. M2 porins. Okay, so this porin helps the migration of protons from host cell to into the DNA. So once this proton migrates, then acidification happens here, and then uncoating happens, and then the genetic material will be released into the host cell. So next class of drugs, okay, that is entry inhibitors. So under entry inhibitors, you have, that is, okay, before cell entry, that is entry inhibitors and uncoating inhibitors. Next one is, once if it enters, okay, that target is over. Next, once if it enters into the host cell, so then what are the drugs we have? Okay, then first one is the reverse transcriptase inhibitors. So this is examples you have. Okay, that is zidovudine, lamivudine. You can write it down, and once you write it down, you can raise your hand, so that till that time I'll wait, then I'll explain you further regarding the reverse transcriptase inhibitors. Okay, if you have written down these structures, okay, first let me tell you about the reverse transcriptase. Okay, reverse transcriptase, it is a viral enzyme. Okay, remember that it is a viral enzyme which is present inside the virus. So then what is the function of this reverse transcriptase? And especially this reverse transcriptase, it is associated with your HIV virus. Okay, that's why HIV virus also is called as a retrovirus. So this reverse transcriptase, it is an enzyme which is present in retrovirus type of virus. And then what is the function of this reverse transcriptase? So we know that viral genome, if you see that viral genome, okay, some virus are be classified based on their genome. So some viruses are called as a DNA type virus. Another type of virus is called as a RNA type of virus. So here the DNA type of virus also, it can be a single strand. Normal in your humans, if you see that the DNA is, or DNA genome, if you see, it is a double stranded DNA. And here if you take the virus, its genome is, it majorly, if you see that it is a single strand DNA or single strand RNA. So this difference you remember. So the DNA, that is why genome present in the RNA, if you see that, it can be DNA or RNA. The DNA also sometimes it will be single strand or it will be double strand, or in RNA case it will be a single strand RNA. So this is the basic difference. Now coming to the reverse transcriptase, it is an enzyme which is present with the virus. Okay, so what is the function of this reverse transcriptase? So the reverse transcriptase, what it does is that it synthesizes DNA, okay, from RNA, okay, complementary DNA from RNA. So now it will become a double stranded. So in retrovirus, if you say HIV virus, so you have their only single strand, that is RNA. And now using this RNA, okay, and host a replication mechanism, that is, what the virus does is that it synthesizes DNA from RNA. Normally, normal process, if you see that from DNA we will get RNA. That is normal process. But here it is opposite happens. So opposite, that's why we call it as a reverse transcriptase. Transcription, we know that. Transcription means synthesis of RNA from DNA. Reverse transcription. Reverse transcription, what happens is that from RNA, DNA will be synthesized. Once if this DNA is synthesized, so that is there the function up here, reverse transcriptase. So reverse transcriptase, what it does is that it synthesizes RNA, that is DNA, complementary DNA from RNA using host replication mechanism. Host replication. So this virus is not having any replication mechanism on its own. It is not having. For its replication, it has to use the host replication mechanism. So by using the host replication mechanism, virus, what it does, like HIV, so virus, what it does is that it synthesizes DNA. Clearly remember this, it synthesizes DNA from RNA. So reverse transcription. Okay, and then, so here, once the DNA is synthesized, okay, once the DNA is synthesized and this DNA is inserted into the host DNA. Okay, once the DNA is synthesized from RNA, now you have a double-stranded DNA. This double-stranded DNA will be inserted into host DNA. You clearly remember this, follow this. Once the DNA is synthesized from RNA, a synthesized DNA is inserted or integrated into the host DNA. Host DNA. So this is what happens. So in the life cycle of virus, we have studied that. So these are the drugs, that is zidovudine, lamivudine. What they do is that they inhibit the enzyme reverse transcriptase, so that it prevents, by inhibiting the reverse transcriptase, it prevents, okay, synthesis of DNA from RNA. And once the DNA is not there, it cannot be integrated into the host DNA. Replication can be stopped, and virus replication can be stopped. So this drug, these are the drugs, and these are called as a nucleotide analogs. Okay, nucleotide, you have here, if you see the zidovudine, you have it is a thymidine analog, and the another one, if you see that lamivudine, it is a cytosine analog. Okay, cytidine analog. So these are nucleotide analogs, because nucleotide, you know that nucleic acid nucleic acid plus sugar, that we call it as a nucleotide analogs. So zidovudine and lamivudine. So these two are the nucleotide analogs. Okay, we have examples, and go to the next slide. So then adenosine. Okay, adenosine is a it is a non-nucleoside, that is not, it is not a nucleoside analog, but it is a reverse transcriptase inhibitor. Adenosine. So here, if you see that, that is a hypoxanthine. It is a hypoxanthine. You can write it down. Next to that adenosine, it is the hypoxanthine. Hypoxanthine it is. So you can see that hypoxanthine is not a nucleic acid. Okay. So the previous drugs, if you see that these two, this is a nucleic acid, and this is also nucleic acid, cytidine and thymidine. So whereas here adenosine, if you see that adenosine, so it is a, here it is a hypoxanthine. Hypoxanthine is not a nucleic acid. It is not a nucleic acid. And here these two structures, if you see, okay, this is the okay, here if you see, that is here, deoxythymidine. Okay, first you write this adenosine. If you have if you have written that, you please raise your hand. If you have written down that structure, okay. So this is adenosine, okay, and this adenosine will have the hypoxanthine structure. Next one is that galcitabine. Zalcitabine. I'll write that. Okay, let me do. This is galcitabine. That also you take. Second one, when you write it down, you just this is galcitabine. Galcitabine. When you write it down, you please raise your hand so that I can go to the next one. So if you have galcitabine, this is also, this is the, here if you see, this is a cytidine. Okay, this is cytidine, and if you see that dideoxycytidine, it is dideoxycytidine. So these two hydroxy, this is cytidine, actually normal nucleotide which is present. Okay, and to this analog we are giving, okay, the reverse transcriptase inhibitor, that is galcitabine, and here dideoxy, two hydroxyl groups are missing. Okay, so these are, this is, and adenosine, okay, and galcitabine. And previously two more drugs we have discussed, that is zidovudine and lamivudine. These are also reverse transcriptase inhibitors. And now that adenosine and didenosine and galcitabine, these two are also reverse transcriptase inhibitors. So can I go to next slide? Okay, I'll go to the next slide if you have taken this. And next, here, these two are the drugs, okay, lovirid and then delaviridine. So these are also reverse transcriptase inhibitors, but these are non-nucleotide reverse transcriptase inhibitors. Non-nucleotide. Previous one of nucleotide reverse transcriptase. Four drugs which we have discussed. Okay, lamivudine, zidovudine, okay, zalcitabine. Okay, so those drugs are nucleotide analogs and reverse transcriptase inhibitors. And these two drugs, lovirid and then delaviridine, these are non-nucleotide reverse transcriptase inhibitors. You take down these two structures, and when you have finished, you just tell me. You raise your hand when you finish that. Okay. Okay, I'll go to the next slide. And so reverse transcript, as I was explaining to you, okay, as I was explaining, reverse transcriptase, it's a viral enzyme, okay, and then this viral enzyme, what it does, as I told you, it generates or synthesizes complementary DNA for RNA template, because RNA itself, RNA already it is there with the DNA, that is virus. So viral RNA will be there. So using that RNA template, it synthesizes the complementary DNA. Okay, and this reverse transcription is done by these three enzymes, biochemical reactions. So the retroviral, that is RTS, these are the biochemical activities we have. Three are there. So the first enzyme, that is which helps in the reverse transcriptase, three common units are there. So the RNA dependent DNA polymerase, so that is first one, RNA dependent DNA polymerase, and ribonuclease, and DNA dependent DNA polymerase. So these three enzymes, finish the process of reverse transcription. All these three, they synthesize DNA from RNA. So reverse transcriptase inhibitors, they inhibit this process, reverse transcription. So the reverse transcription is synthesis of DNA from RNA. Okay, so this RNA is present with the virus. Okay, and then from this RNA, DNA will be synthesized. So double stranded DNA, and this DNA is inserted into host genome, host DNA. Okay, so that is what happens with the reverse. So you give the reverse transcriptase inhibitors. So these reverse transcriptase inhibitors, they inhibit the reverse transcription, thereby prevents the synthesis of DNA from RNA, so that the virus cannot insert the DNA into the host DNA, and replication can be stopped. So these you don't take, these are a previously also I have given you, previous slide also, don't take these structures. Examples I am giving you, nevirapine and etravirine. So these are non-nucleotide, reverse transcriptase inhibitors, other examples. Next class of the drug. So next target for us is that, suppose DNA is synthesized. So here it is. First the virus attached to the host cell. Then to prevent that, there we have used entry inhibitors. Suppose it is diffused, then it has to release its genetic material into the host cell. So there we have used uncoating inhibitors like amantadine, amantadine. Once if it is released, then they have to synthesize the DNA from RNA. If you have taken down, listen to me. I'll take it down, then I'll explain you. So if you have taken down, and you raise your hands. Please raise your hand, because our time will be 40 minutes. Okay, I assume that you have finished writing the structures. So now coming to the topic is that integrase inhibitors. So integrase inhibitors. So it is, the case is like that. So we have seen the virus life cycle. In the virus life cycle, if you see that, first it binds GABA host cell, and that we call it as that is where we have seen that entry inhibitors, and once if it diffuse into the cell, then it has to release the genetic material into the host cell. Then we have seen that uncoating inhibitors. Then once supposing that their genetic material is released, viral genetic material is released into the host cell. Okay, and then there we have seen that DNA synthesis, okay, or reverse transcriptase working and synthesis of DNA from RNA and this. And once if it is synthesized, next step, what is the next step? So RNA synthesized is DNA from RNA. Then what is the next step? So next step is that the synthesized DNA, okay, from RNA, that has to be put into the host gene. Host genome, it has to be inserted into the host gene. That means we, a human, suppose HIV virus is there. HIV virus, what it does is that it synthesizes DNA from RNA. Then the synthesized DNA will be inserted into the host, that means human's DNA. So here the insertion is of viral DNA into the host DNA is done by the enzyme called as integrase. Okay, integrating the viral DNA into the integrating the viral DNA into the host DNA is called, that function is carried out by integrase. So once if it is integrated, okay, listen here carefully. Once the viral DNA is integrated into the host DNA, then what happens here? So now the viral DNA is present in the host DNA. Okay, so now the host DNA considers viral DNA as its own DNA, and then transcription, translation, all the things will happen. Okay, so once the viral DNA is inserted into the host DNA, then host DNA, okay, will consider, our host will consider the viral DNA, okay, as its own, and then transcription or translation process will happen. Then the RNA genome what is required, and the protein code what is required, or the capsid what is required, or the enzymes which are required for the virus, all will be synthesized from host DNA, because the viral DNA is inserted into the host DNA. So all the components which are required for the assembly of viral particle, what are the components that are required for the assembly of virus particle are synthesized. When they are synthesized, once the viral DNA, that is, once the viral DNA is inserted into the host DNA. Once the viral DNA is inserted, now the viral DNA will become a part of host DNA, and the host DNA or host will consider viral DNA also its own DNA, and that DNA genome, that gene also, viral gene also will be expressed. Okay, the viral gene generally you will have a that is lipid layer, or you have the capsid layer, or you have the enzymes reverse transcriptase or integrase or protease. All these enzymes are synthesized by using the host, okay, that is a synthesis mechanism. So this is happens. Now integrase, what it does, integrase function we already learned. The integrase function is that insert the viral DNA into the host DNA. So now this step you can prevent by giving the integrase inhibitors, that is raltegravir and elvitegravir. Okay, these two drugs you can give integrase inhibitors, so that the viral DNA will not be incorporated into the host DNA. So thereby you can stop the replication of virus. So I'll go to the next slide. So integrase is a viral enzyme which inserts the viral genome into the DNA of the host cell. That is just the function of viral integrase. Next, protease inhibitors. So you can take down the structures indinavir, sacubitril. So you just, once you finish taking these two drugs, you just raise your hand so that we can proceed to the top. I'll explain you. So once you write down these structures, Okay, do you have, so better you write down the structure so that you will get the practice for your exam. So don't sit idle. So once you write it, you give me, raise your hand so that I can proceed to the next slide. Okay, so I hope you have taken down and I can prove. Okay, so now listen to the thing here. Okay, protease. So protease, if you see that, okay. So next, see the protease inhibitor. So protease, it is a viral enzyme again, like reverse transcriptase. It's a viral enzyme which is required for the synthesis of DNA from RNA. Then we have the integrase. Integrase, again, it is a viral enzyme which is required for the integration of viral DNA into the host DNA. Then protease. Okay, so protease also, it is a viral enzyme. Okay, so which is required for converting immature, that is processing the protein. So there won't be any proofreading here. It is only processing, slicing, okay, slicing, okay, of the protein. So the protein will be, okay, the protein will be made into immature, that is immature protein will be converted into a mature protein. So the protein here means, so can be its enzymes, okay, here, or glycoproteins or whatever the things. So here the protease enzyme, what it does is that it converts immature protein of viral, that is synthesized viral proteins, into mature viral proteins, mature viral proteins. So the how it is done, by slicing the unnecessary parts, okay, of the protein. Unnecessary parts of the protein will be sliced off. Only necessary part of the protein is kept. Necessary part of the protein is kept, and thereby immature protein will be converted into a mature protein, that is, which is, okay, the protein which is not required will be converted into a required protein. Okay, required protein. That's what is done by the protease enzyme. Okay, so we know the steps. Okay, we know the steps of virus life cycle. So entry, then once the entry is over, then uncoating, and uncoating is over, then DNA synthesis. Once the DNA is synthesized, then the DNA will be integrated into the host DNA, and once if it is integrated into the host DNA, so virus requires its capsid, it's a lipid layer, and it requires its enzymes reverse transcriptase. Uh, it requires its integrase enzymes. All these are synthesized by using the host mechanisms, and of that, one is the protease enzyme. So once the proteins are synthesized from the host using the host DNA, these immature proteins will be converted into mature proteins, so by enzyme protease. So we are giving here protease and enzyme inhibitors, indinavir or sacpinavir. So they inhibit the protease. Okay, so thereby we stop the conversion of immature into mature. So the proteins will remain as immature only, so that they will not be useful the virus. So then I'll go to the next slide. So this is another example, ritonavir. Ritonavir, this is also protease inhibitor. So once you write down the structure, just remind me. Okay. So this is about the protease inhibitors. Okay, 3 drugs we have discussed of the protease, and so first to the indinavir and sacfinavir, and other one is that ritonavir. So this is next. Okay, so here I stop the class here. Okay, and we'll continue with the next class tomorrow. And if you have last 5 minutes time is there, almost 5 to 6 minutes time is there. Okay, so if you want to have any questions, you can ask me. Okay.